The battle never truly ends for survivors of childhood cancer. Over the next few minutes, I'm going to be exploring the repercussions that cancer treatment can have on the brains of childhood cancer survivors. My name is Karthik Reddy, and I'm a current undergraduate student in the lab of Dr. Donna Murdaugh at the Children's Hospital of Alabama. Our project explored a cancer called acute lymphoblastic leukemia, or ALL for short. This is one of the most common pediatric cancers. Despite its high occurrence, this cancer has one of the highest survival rates due to current chemotherapy treatments. But these treatments have big drawbacks. Chemotherapy drugs like methotrexate, which are used in the treatment of ALL, have been shown to have toxic effects on the developing brains of children. Once the chemotherapy is over, many patients experience what are called cognitive late effects, which are impairments in cognitive abilities. One specific area shown to be affected, and the focus for our study, is called working memory. This is our cognitive ability to, in the short term, store and manipulate information. You may now wonder why this specific area of cognition is so important. It's because ALL largely affects children. ALL patients already have significant delays in cognitive development due to the disease, as well as chemotherapy-induced neurotoxicity, which is toxicity that affects the neurons of the brain. Working memory is a crucial component of early childhood learning and critical thinking, so any deficits in this area are a compounding factor that can further disadvantage ALL survivors. Despite the importance of this issue, very little work has been done to understand what exactly is occurring on a neurobiological basis within the working memory network of ALL survivors. Our study aims to address the needs of pediatric cancer survivors by examining associations between neuropsychological assessment of working memory and functional magnetic resonance imaging, or fMRI, in order to better understand the cognitive late effects of chemotherapy treatment. We hypothesize that ALL survivors will show reduced resting state functional connectivity of the working memory network that will be associated with reduced performance-based self and parent-rated working memory. You can now pause the video to see the demographical breakdown of our participants. Several of our ALL participants had to be excluded due to movement during fMRI. We used 6 ALL participants and 12 control participants for the study. As I mentioned previously, neuropsychological measures were used to assess working memory. Our objective, performance-based measure of working memory, was an administered subtest of the Wechsler Intelligence Scales. The subjective measure of working memory was obtained from the brief. This is a rating survey given to the participant and the parent. The questions of the survey are only correlated with working memory, and you may pause the video now to see examples of the subjective questions found on the brief. We then compared the neuropsychological results between the ALL group and controls. First, keep in mind that a higher score on the brief measures, which are the self-rated and parent-rated working memory subscales, indicates a poorer perception of working memory abilities. When you look at the first 2 bars, you see that the working memory abilities of ALL survivors were rated as being subjectively worse than controls. Yet, the performance-based working memory yielded no significant differences between ALL group and controls. This perceptual gap of lower working memory ability in ALL survivors may indicate neuroplastic adaptations that are occurring to compensate for any neurological damage from the cancer treatment. This could be a possible rationale for why they perform similarly to controls in performance-based measures of working memory. To verify this hypothesis, we acquired resting state fMRI scans, which refers to the activity among brain regions during rest, or the absence of a task. Using Neurosynth, we generated a meta-analysis of key working memory regions. We selected 4 of the regions of interest, also called seeds, to use in our analyses. These include the middle frontal gyrus, the inferior frontal gyrus, the cerebellum, and the inferior parietal lobule. First, we looked at ROI to ROI analyses, which means connectivity within our identified network. No significant differences between the groups were found. However, there were still variations in the intensity of connections between regions. A darker color indicates higher resting state functional connectivity between regions. Next, we looked at seed to voxel analyses, which is looking into the connectivity of a specified region within the working memory network to the entire brain as a whole. There were 2 main significant results. In ALL survivors, we found that the resting state functional connectivity of the middle frontal gyrus to the occipital cortex was significantly correlated with performance-based working memory results. In controls, we found that the resting state functional connectivity of the entire working memory region to the supramarginal gyrus was significantly correlated with parent-reported working memory. In conclusion, we found that both ALL and control participants have intact working memory brain networks. Our within-network ROI to ROI analysis demonstrated that, while not significant, there were still differences in working memory regions between ALL survivors and controls. Future studies with a higher sample size could show significant differences. Our seed to voxel analysis of ALL survivors may indicate that they are recruiting regions beyond ones involved in working memory to perform similarly to controls on tasks requiring working memory function. The neuroplasticity, or the ability of the brain to adapt to damage, is especially high in children and could provide rationale for this conclusion. The results of this study can be used to create additional treatment options for survivors to not only survive, but thrive. Future pharmaceutical and psychological interventions can be tailored around these possible compensatory mechanisms that are hiding deficits in working memory function among ALL survivors. The battle with cancer never truly ends, but this study is a step in the right direction. Here are our references, and I'd like to thank the following individuals and organizations. You may pause the video now to look at my poster in more detail.